Okay, thank you. I'm Rick Stevens, and today I'll be talking about high performance computing and AI in COVID research. And I'll give an overview of what we're doing in the US. Of course, I can't talk about the whole effort nationally. I can talk about what we're doing in my lab and something globally globally in the US about the HPC consortium. So earlier this spring, right after the pandemic was getting into full swing, there was a proposal started by IBM and then rapidly gathered by a lot of institutions to stand up a high performance computing consortium to support the COVID-19 related research projects. And currently, this consortium is still going. There are 66 active projects, and together the resources that are part of this consortium are over 480 combined petaflops. And the research that this is supporting is covering everything from really basic science about the virus, about proteins, and about drugs, all the way to trying to optimize things like supply chains for PPE. The combined resources of this consortium include over 5 million CPU cores, over 136,000 nodes, over 40 members are either contributing cycles or resources of some form. We also have, in addition to the supercomputer centers, we have allocations on the major cloud providers, Amazon, Google, Microsoft, and there's over 50,000 GPUs that are combined in this set. So the consortium has a governance board, which I'll talk about in a second. So there's industry members from companies, there's national labs, mainly providing cycles and reviewers. There's university groups providing either computational resources or, in some cases, reviewers, and resources from the National Science Foundation. But the consortium also has a linkage to the EU PRACE COVID initiative, and in that sense, they're coordinating across the Atlantic. There's a board that I mentioned that's governing how this resource is being used. It's chaired by Paul DeBar from DOE and Dario Gil from IBM. Barb Halland, you need to know, is executive director, and it contains representatives from each of the groups. And Jim Brassi from Livermore has been really instrumental in standing up the whole infrastructure, really Jim and Mike Rosenfeld from IBM. And OSTP has also been very supportive of this. So the way this works is that individual groups can make proposals to the consortium. They're reviewed kind of once by a panel that reviews both the science and the technology needs. Then they're the centers are asked if they want to support this, and they do a matchmaking exercise, and then a center makes an allocation. And so the advantage of this, instead of individual projects having to talk to each center separately, they only have to write one proposal that's looked at collectively. And it's been quite successful. There's 66 current projects. They're in 3 broad categories: basic science, covering things like the virus structure, function of proteins, protein interactions between the virus and human, environmental effects, and viral evolution. Therapeutics is the largest group, basically drug discovery, antibody design, discovery, design of proteins, small molecules, and drug repurposing, computational activities and drug screening, and things like that. And then patient care things, really things more related to optimizing the healthcare system or epidemiology. And to give you a sense of the flavor of some of these projects, I just highlighted a few here. The first one just randomly came up is this a molecular dynamics study of the SARS-CoV-2 helicase protein, the NSP13 protein, which is actually one of my favorite drug targets. And I was looking at this and I noticed the name, and it's a person called Stephen Rick, which is of course not me. Has almost the same name with my first and last name reversed. That's what I look like. I don't know what this person looks like, but they're at a different university, but it's basic science. But you can see a lot of molecular dynamics, designing inhibitors for the spike protein, accelerating research using machine learning, multi-scale models, including a proposal one of my team members, Argonne and partners are doing that includes modeling the whole virus. And projects like understanding cell type specific infection paths and a lot of AI. So I think it's a broad collection of projects from the community. So before I go on to the more detailed discussion of what we're actually working on, I thought I'd give some overviews on the virus. So if you're going to work on this problem, you need to kind of understand the enemy. And here's a kind of a summary. So SARS-CoV-2 is the virus. Of course, the disease is called COVID-19. The virus is small. It's 100 nanometers. It's got a very tiny volume, 10 to the minus 3 femtoliters. It's a single-stranded RNA virus, meaning the payload inside is just RNA. And it's decorated, this little sphere is decorated with these spike proteins, and these spike proteins interact with receptors on the human cell, and that's how the virus gets into the cell. It turns out that that the ACE2 receptor that this thing needs to get into the cell is a good antibody target because it's exposed and you can recognize it outside of the cell. There's also a protein, a human protein on the cell surface called transmembrane protease, serine protease 2. And this is a really interesting drug target as well. The RNA is wrapped around these little proteins called nucleoproteins, or protein N, and that's also potentially a drug target. Just so you have a sense of how this virus works is that after infection, it takes about 10 minutes or so for the virus to get into the cell. It latches on to this ACE2 receptor and kind of gets pulled into the cell. It's in the cell for maybe 10 hours. During that process, it breaks open, the RNA comes out, it starts to get processed by machinery and starts to make copies of itself. And after about 10 hours, virus particles will start exiting the cell, and each infected cell produces about 1,000 copies. And these are rough numbers from culture studies that people have done. We actually know the protein structures for most of the proteins. The virus RNA codes for about 30 proteins, 27, 28 proteins, in a very interesting fashion with some very long polyproteins that have to be chopped up using proteases that are actually encoded in the genome itself. In this map here, these are structures that were modeled from the original SARS-1 virus and then tweaked up as we started getting sequences for SARS-CoV-2. And what you can see in purple here are mutations from SARS-1 and from the areas that are different, say, between the SARS-CoV-2 virus and the nearest related virus, which is from a bat. And the yellow is places where there's new genetic material. And you can see that the spike protein, this big protein on the left, has a lot of mutations and some novel inserts, and that's necessary for it to actually enter into the human. Now, each of these proteins in principle is, in some high-level sense, is a potential drug target. Over the last couple of months, there's been a huge amount of activity in producing new structures, and here's some cartoons of those structures and the PDB database entry that's associated with those. A lot of these were produced at the Advanced Photon Source here at Argonne and at other light sources around the world. And many of these are actually quite interesting drug targets. About 2/3 of the proteins are used in commandeering the host cell machinery and helping it produce copies of the virus, and about 1/3 of the proteins, of the ones on the right-hand side, so-called structural proteins, actually are used in packaging up the RNA and producing the virus itself. Um, Here's a kind of a map. So on the left-hand side, these are the list of proteins, the similarity between SARS-CoV-2 and the original SARS-1. And most of the proteins are, you know, 95% similar or more. A handful are less so. And the things that I've tagged here with green are structures produced at the light sources supported by DOE. And on the right-hand side are these blue stars, which are plausible drug targets that we're trying to go after. On the right-hand side here is a diagram at the top. It shows viral entry, the stages, and a handful of drug targets that can be inhibited to block viral entry. In the middle is a set of processing for viral replication and post-processing. And then there's some targets that are in the host that might kind of interrupt host processes that, that the virus manipulates to either make it more hospitable or where it kind of attempts to turn off the immune system or the innate parts of the immune system. One of the key opportunities with this, of course, is to use large-scale computing to model the function of the proteins. Our team, this is work led by Arvind Ramanathan and collaborators at Stony Brook, Carlos Simmerling and Shantanu Jha, who is at Rutgers and at Brookhaven, to do large-scale simulations of the interaction between the virus spike protein and host, the cell, the human host receptor, the ACE2 receptor. And you can see in this simulation some interaction between the spike, which is the one in yellow and blue and red, and the host receptor on the cell. And in nature, what happens is this thing will interact and then it will get pulled into the cell. And what we're trying to understand here is how this interaction actually works and what part of the spike protein might be a suitable antibody target. So you could design an antibody that would fit right on there and recognize it. We're also interested in using high performance computing to make the best out of all kinds of information we're getting about these structures. So this is an example here of doing coarse grain work. So we have models of various proteins coming from light sources and coming from electron microscopy sources. In this case here, cryo-EM image. We can do a finite element kind of coarse grain model, and we can do a refined atomic level precision model of the virus. And high performance computing is being used to basically go back and forth. We're using AI methods to actually do the interpolation between these two systems and to constrain them to biophysically real motion and so forth. So the virus also interacts a lot with human proteins. There's a number of papers that both are trying to document this from experimental data and from kind of inference data from biological networks. Maybe up to 300 or so proteins have some kind of interaction. The reason that this is important is we want to look at, for example, on this chart here, the proteins in the middle, the red stars are viral proteins, and the little diagrams here show host proteins that they are predicted to interact with. And in some cases, we can do pull-down studies and actually have experimental confirmation that they interact with them. The proteins that they interact with are potential drug targets depending on what they do and depending on if we have reasonable inhibitors or could design inhibitors for those targets. And if inhibiting those functions would disrupt the virus life cycle, but actually not injure the host. Part of our effort has been to combine AI and simulation, and early on in the work this past spring, the team at Argonne started assembling a very large database. It's about 60 terabytes of data from over 4 billion compounds, and it includes things like drug descriptors and images and fingerprints for each of these molecules that are essential for using this data in machine learning applications. So one of the strategies that we have is to use a combination of high throughput virtual docking, what you think of as virtual screening, to generate scores, that is docking scores, how well a molecule might interact with a protein, might dock with a particular receptor. We generate that on thousands or millions of data points, sometimes randomly chosen from the libraries, sometimes in very specific libraries, but then use that data to train machine learning models and then do inference on a much larger set. So this idea of physical computation, producing a data set, training an AI, and then sweeping AI across the large scale data is one of the strategies. We also use things like neural fingerprints to make searches and so on. And from that, we can get enrichment of the set of molecules that are likely to interact. And here's kind of a diagram that kind of ties this together. We get structures from the light sources. We do modeling, molecular dynamics modeling. We have data coming from literature, data coming from drug databases. And we use this hierarchical process for screening and producing hits. We then do more detailed modeling to refine that, and the ultimate hits get passed on to wet lab screening. Early on, there was a big database put together by the community, academic community, literature. It's some kind of large-scale 45,000 article database, and we've been using natural language processing methods to comb through this to try to augment the set of known drug interactions with the molecules and use that to prioritize some of our drug targets. Early on, we focused on looking at repurposable drugs, so drugs that are already in the marketplace. There's about 4,600, about 2,600 drugs that are worldwide already licensed by national authorities, and another 7,000 or so that are in various pipelines, and we've been screening those. These are really important because if they show activity, they could be put into clinical practice relatively quickly. Here's some example targets and some example high scoring hits. These are not in any particular order, they're just kind of the top chunk. And all of these have been ordered and are in our various wet lab assays for validation. And our goal for validating some of these is to move from kind of computational hits to what we call advanced leads. That would be molecules for which we have some experimental evidence that they are active on the target, and then those would get further refined downstream. Here's an example. On the left is a molecule from the zinc database. That's one of our highest scoring hits on this particular target, methyltransferase, which is also called the non-structural protein number 16. And you can see the binding site and the molecular structure here. And on the right is the highest scoring kind of existing drug, an experimental drug. As you can see that the non-drug compound in this case scores quite a bit higher and probably has higher potential than the existing repurposing drug. But it's important to look through all the existing drugs and to test as many of those as we can as we move, and then also to move on to novel compounds. We're using various kinds of computational modeling to understand how when you dock a molecule to a protein, does it change the dynamics of the protein? Does it do what we think it does? Does it stay in the active site? Does it get tossed out? This is a particular drug target called an ADRP target, called NSP, also known as the non-structural, as part of non-structural protein number 3, and you can see on the right compound that kind of stabilizes this molecule compared to the one on the left. We're using machine learning to actually analyze the trajectories of these molecular dynamics simulations and to categorize the state so we can understand whether or not a drug causes the molecule to change states or what it stays in a given state, and how likely that state is to be from a frequency standpoint in actual biophysical conditions. One of the overall challenges here, of course, is there's over 10 to the 60th possible drugs, and you can only test at the end of the day in humans, a small fraction of these. And so it's a big funnel. And one of the things that our group has been working on, particularly my PhD students, Austin Clyde and his collaborators at the university, have been building a reinforcement learning-based system, and you can see this molecule up here in the middle that's kind of growing and adding things on. The idea with this system is that you have a molecular dynamics model of the protein. You start with an initial compound in the binding site, and then it uses reinforcement learning to grow that molecule along potentially synthesizable directions. and simultaneously trying to optimize its binding energy and other properties like toxicity or wherever we have models for. And this is an example of putting this into a reinforcement learning context, combining molecular modeling, biophysical modeling, and machine learning. So I think this is the future. This is the future kind of direction. And in some ways, the current crisis has allowed us to put a lot of resources into imagining what these future systems could be like. And of course, these are going to be useful for all kinds of drug design problems going forward. We've gathered tools and models and data, and here's some of the URLs for this system. We're very grateful for all the computing time that we've been able to get at Argonne, Oak Ridge, at TAC, using the Frontera machine, resources at SDSC, Comet, and other machines, and we recently were were able to get some of the earliest DGX3 machines with the new A100 NVIDIA GPUs. They've been really amazing, and we also have collaborations with RZ and other places. So we're very happy about all the access. So, and to end, this is a large team. Early on in March, we put the Slack channel together. We started gathering people, and within a few months, we had over 250 people involved in this. I can't say everybody, but certainly Ian Foster and Robin Opp and Tom Breton, Austin Clyde, and some of Ian's team, like Kyle Chard and so on, have been instrumental in getting this work done. And so with that, I thank you.